Hello, everybody, and welcome to another episode of Meet the Researcher. Today, I'm excited to welcome 2019 VHLA Research Grant recipient, Dr. Anna Matynia. Dr. Matynia is an associate researcher at UCLA, where her work involves better understanding retinal function, diseases of the retina, gene expression in retinal cells, and retinal tumor formation associated with VHL. Dr. Matynia, thank you for joining us today.
My name is Anna Matynia. I've been doing some research in the VHL arena for the past 3 to 4 years. And we started this work because I work with Dr. Michael Gorin here at UCLA in the Jules-Stein Eye Institute. He is a clinician and physician who has treated VHL patients for over 20 years now, starting when he was at NIH. So he still sees a few of these patients in his clinical practice. And so I've gained a lot of insight about the clinical picture of this, but also we've explored together and discussed together what exactly is happening in these patients and what are some things that can be done for it. Because for the longest time now, the only thing that, and even currently, the only thing that can really be done is to treat these patients that have retinal hemangioblastomas with laser cryotherapy to try to stop the damage from these tumors, but it certainly doesn't cure it, and it certainly doesn't prevent it. So I work in mice. I don't work in people, but we use mice to model what's going on in humans. And the first thing that you have to do is to be able to develop a model that works in mice, so that and that mimics the human condition. So with that, I'm going to start my presentation, and I will come back at the end with the live view. So let's see. Here we go. So, so we started with the idea that there really wasn't a good model in mice that formed these benign tumors that occur in people called retinal hemangiomas or retinal hemangioblastomas. So, as I mentioned, VHL is a tumor suppressor gene. Well, it's a cancer predisposition. VHL disease is a cancer predisposition disease, and it's because VHL is a tumor suppressor gene. So it tries to keep cancer from happening. However, when you then get a mutation in it, this ability to suppress tumors now is abrogated. So people with VHL usually inherit one mutant copy of VHL at birth, and about 20% of patients have a de novo mutation, meaning that their parents did not have it. Most tumors, if not all, we say most because we don't have absolute on this in terms of surveillance, but most tumors show what's called loss of heterozygosity. And what that means is that the person acquires a mutation in the second copy of their VHL gene. And just down below here, I've shown some genomic Southern blot data showing that you have lost both alleles. So if you had a single allele, you would see a band in both positions. If you have loss of both, you would see none. If you had only one loss, this would be lighter than the control. So T stands for tumor, N stands for normal. And so you can see in a number of patients, you see a decrease or a loss of the second copy of VHL. So we also know that this only affects specific tissues, or predominantly affects specific tissues. The disease ends up with both non-malignant and malignant tumors that affect these different tissues. Kidney is a big one where you get clear cell renal carcinoma, the adrenal glands with pheochromocytomas, inner ear endolymphatic sac tumors, and benign tumors of the reproductive tract. I hope you're seeing my cursor. So this is just a schematic on the body of where these tumors occur. So the place that I have interest in is the tumors that form in the central nervous system, or the CNS. And this includes the brain, the spinal cord, the retina. Technically, spinal cord's not the CNS, but these hemangioblastomas form there as well. So We are in a department of ophthalmology, and our best abilities are in the retina, which is the central nervous system. It is directly connects with the brain, and it is actually a really good tissue to do a lot of studies in because it's accessible. We can image it in both people and mice live. We can do in vivo imaging, and we can follow that across time. And then, of course, when you use models or on surgical samples, you can then look at these ex vivo in pathology. So I'm going to show you a picture of what this looks like in people. So retinal hemangioblastomas are typically large and highly vascular. In this fundus imaging down here, you can see the tumor down below in the lower right-hand corner of the image. And what you see that is a lot of these very large dilated vessels. These are called feeder vessels that are feeding the tumor. You can see this on a fluorescence angiogram. So this is just, fluorescence retinogram. This is just like when you have an angiogram for your heart, you can see the vasculature. So you can see the fluorescein in the vessels, you can see the tumor here and some dilated vessels that are feeding it. And this is in contrast, there's like a small tumor here. And so these can become very large. They're typically located in the superficial retinal layers and project into the vitreous. And they themselves are benign. So there's no real risk of metastasis and cancer in that sense. However, they can grow large enough to, one, block vision because they just take up space. And second, a really significant consequence is that you can get retinal hemorrhage from these tumors. So, and exudation. So these retinal tumors become highly vascular, and they aren't the normal tight vasculature that you see in the rest of the retina. It allows fluid sometimes to pass out of the retina into the surrounding out of the vasculature into the surrounding retinal tissue, and that's exudation. The other thing that happens is that sometimes these vascular tumors grow into the vitreous, and the vitreous actually moves in your retina. So this can cause some shearing and some breaking and bleeding. So it's a retinal hemorrhage and exudation that can lead to vision loss, and if they're bad enough and enough of them, to actual blindness. So there was a lot of work done by C.C. Chan and on the pathology on surgical biopsies or surgical tissue samples of these tumors back a long time ago. And it was really gave us the ability to look at these tumors a little bit closer up, because in vivo imaging is really great for following patients, but it really doesn't give us that good of a view of what's going on. So, so the studies from 1997 and 1999 did a lot of pathology looking at what these tumors consisted of, because we need to know what they're consisted of in order to understand how they happen and how to treat them. So they were described both in the retina and in the brain. So the findings were essentially the same, was that these tumors were highly vascular. I've labeled a V for the vessels here, a couple of large ones, and then a whole bunch of smaller ones, including some other smaller ones that I haven't labeled. You can see, in fact, in this vessel, you can see some red blood cells. So there was a lot of vasculature in here, and this is abnormally vascular for retinal tissue. But it also had a lot of area, that was described as stromal or having giant foamy cells. And these cells were of unknown origin. So these are the two predominant features of the retinal hemangioblastoma. Subsequent studies also showed that these tumors express a number of different genes, including VEGF, CXCR4, HIF, and ubiquitin. These are proteins or genes and proteins that are indicative of angiogenesis, of hypoxia, and immune cells. So this tumor is growing like many tumors. You have tumor mass that grows, but it needs to be fed. So it needs to receive blood, oxygen, energy, glucose in order to stay alive, like all of our tissues. So one of the questions that happens here is this really a tumor of the vasculature, or is the vasculature, the angiogenesis, a response to the tumor formation? And that question hasn't really been fully answered yet. So one of the things about doing pathology on patients and excisions from patients is that these are fully developed hemangioblastomas. So it tells us what happens in the late stage, but it doesn't tell us how they form, what initiates formation. And in fact, I already kind of told you that these cells are of unknown origin. So this has already become a cell that we don't really know what it was. So it may have changed across the course of disease. So I'm going to now, sorry, there we go. Okay, sorry. Go over what the cell types are in the retina. And so what the cell types are and what cells might be involved in the tumor formation. Because if we know what the cell types are, then maybe we can later target a therapy towards those cell types based on what they do and what they are sensitive to. So I have over here on the left-hand side of the screen, this is a cross-section of the retina. It's a really pretty section where you can see the nuclei stained in purple. And this shows you the architecture of the retina, of the neurons in the retina. So in the lower layer, these dark, densely packed neurons are the nuclei of the rod and cone photoreceptors. The next layer of neurons that are a little less dense stained and a little darker, a little lighter, are intermediate neurons that process light reception from the photoreceptors and transform it as it goes through the retina. And the very top layer are these retinal ganglion cells. So they receive information that's been modulated from these inner retinal neurons. And these neurons at the very top are the retinal ganglion cells that form the optic nerve that sends information directly to the brain so that we can see and do many other things. So those are the retinal neurons. Right below the photoreceptors, not shown in this image, but cartooned here, are what are called retinal pigment epithelial cells. So these line the edge of the neural retina, and they then, underneath these cells, is a large vascular supply. So this is sort of the boundary between the central nervous system tissue of the retina and then the circulatory system in the eye. So this is the more general tissue of the eye. So these cells have a very important role in protecting the retina from invading certain things in our blood vasculature from below. Now, there's also blood vessels in the retina in this top layer that go and feed the top of the neuron. So the retinal pigment epithelium and the blood flow below supplies the retinal neurons, the photoreceptors. But through the top layer and the intermediate layer, you have blood vessels that go throughout it. And this blood vessel is actually very well built in order to prevent, again, things from the general circulation from coming in and damaging retinal tissue. This is part of what's known as the blood-brain barrier, but I won't talk about that too much at all, other than to say you have the blood vessel here. That's the lumen of it. And then you have the cells that line the lumen of the blood vessels, called vascular endothelial cells. You have another cell type, a pericyte, that surrounds that. And then you have these other glial cells that surround that. So it's a very layered, protected area that mediates and carefully regulates what goes from the circulation into our retina to keep us safe, our retina safe, and keep our vision safe. There are also astrocytes in the retina, and they also actually wrap around blood vessels and do other functions. They're a glial support cell. And then there's these microglia, which is the resident innate immune system of the retina. So one of the things that we were thinking about is, what is a cell type that initiates tumor formation? Well, this cell type should have the ability to either divide in order to find the tumor, or to take a cell that's already post-mitotic or has already given up cell division, but is able to de-differentiate, so go from what it is to a more plastic, earlier cell type, and then redifferentiate into the cell types that you find in the tumor. So it should have some capacity for that. Retinal neurons are considered post-mitotic. They don't have this capacity. The retinal pigment epithelium has this capacity. Vascular endothelial cells have this capacity. Astrocytes really don't have it. I put a check mark because you can take them out and put them in culture and get them to grow, but they're not very good at it. And then microglia also have this capacity. In fact, that's part of the response. So both vascular endothelial cells and microglia respond to signals that cause them to proliferate and to move sometimes. Microglia are migratory cells. So the other thing is that these cells are expected to require loss of heterozygosity for tumorigenesis. Okay, so we're not the first people to think about this. People thought about this for a very long time. Let me just see if that's for, okay. So they've thought about this for a very long time, and they've looked at these cell types, and they have gone in and modified single cell types to delete the VHL gene so that you get loss of both alleles of VHL that should cause a tumor. And they can use a genetic modification using what's called a floxed allele, and I'll describe this a little bit more later, that allows you to remove the gene in a very specific cell. That cell has to be expressing Cre recombinase. And you can do this by using cell-specific promoters that drive expression just in rod photoreceptors or just in RPE cells. And a number of people have done this already. So in retinal neurons, in both rods or retinal neuron precursor cells, if you delete VHL, you end up killing those cells. You get neuronal degeneration, but not tumors. If you use a promoter for melanocytes, which is specifically found in the RPE and not really other cells, not in other cells of the retina, you end up with embryonic lethality. So that means that these mice pups die in utero. If you delete it in vascular endothelial cells, that also ends up in embryonic lethality. Shows you the importance of these cell types. If you delete it in astrocytes, you end up with vascular proliferation, but not a tumor, not something that looks like a hemangioblastoma. And we haven't really looked at microglia. So I'm already pointing you to what we're going to be looking at. So given the ability to proliferate and de-differentiate, we're really considering the vascular endothelial cells and the microglia. Both of these cells also have some evidence in patient tumor histopathology, right? So we saw that there was a proliferation of vasculature. So these vascular endothelial cells, when angiogenesis signals are received, they proliferate, they grow new vessels. So these are clearly involved in the tumor. The other thing is the resident immune cell, the microglia. These basically phagocytose or eat cells that are diseased or damaged. So these could become a giant foamy cell. A giant foamy cell is a characteristic description of a macrophage cell that is surveilling the area and phagocytosing damaged cells. So these two cell types look like they have a role in the final pathology. Okay, so just playing back on the second hit, the first hit. So patients have one inherited variant of VHL, and they acquire a second hit that results in loss of heterozygosity in the tumor cells. So when C.C. Chan and others looked at loss of heterozygosity in tumor cells, they found that that occurred in those giant stromy cells, giant foamy cells of stroma or stroma-like cells. Sorry, I'm going to make a new short form for that, stromy cells. So they found loss of heterozygosity, so they saw deletion of both copies of VHL in those giant foamy cells, but not in the vascular endothelial cells. So this, to us, was a really critical piece of information that perhaps it's not the vascular endothelial cells that are the tumor, but they're just responding to a tumor growth in the retina, much like you get angiogenesis in any tumor that forms anywhere in your body. So maybe it's these giant foamy cells of unknown origin that are the cells of interest. I already mentioned that if you get rid of VHL in vascular endothelial cells, or RPE, they are embryonic lethal in mice. Same thing if you get rid of the VHL gene. So the VHL gene is very important for development. If we don't have it, we're just not going to make it through as an embryo. However, what we can do in mice is we use this floxed allele. So here I'm denoting it with a VHL FL. And again, I'll show you a bit more about that. I'll describe it a bit more in detail later. So we can use these VHL floxed mice that are normal. So they get born in normal ratios, they grow up, they're healthy. None of the tissues that have been looked at that are affected in VHL disease are abnormal in these VHL floxed mice. So what we can do is we can induce homozygous loss of VHL to follow hemangioblastoma formation in individual cells. And we can do that in the adult mice, not in the prenatal or before fertilization and embryogenesis. So that was our goal, was to develop a mouse model of retinal hemangioblastomas in VHL disease and to identify the tumorigenic cell type. The ability to do this in the retina gave us these capabilities of looking at retinal vasculature, both in retinal section, so in histology, and in vivo imaging, using the same sort of in vivo imaging that we use in patients and in people. And it also gives us the ability to assess different cell types in the mouse, and we can do that with retinal sections and with some more whole tissue sort of methods. Okay, so here's that VHL floxed that I told you I'd describe a little bit more. So the VHL floxed mice, these have what's called a LOX-P site, which I've denoted here. And this is just a small piece of DNA. It's 18, 22 bases, something like that, that you can insert into anybody's genome and then make mice, in this case. So what these LOX-P sites let you do is to allow recombination between the two LOX-P sites. So what they will do when you have Cre recombinase there is that they will bind to each other, and the Cre recombinase will cut that loop out. So here we have the VHL gene just schematized. So here's exon 1, exon 2, and exon 3. So we put these LOX-P, we didn't, somebody else did this a very long time ago down here, the paper, right? So a very important gene, people did this a very long time ago because it's very critical to understanding VHL disease, but also development. So they put this LOX-P site flanking exon 1. So that's where you get floxed from, it's flanking LOX-P sites. So by putting the LOX-P sites here, you can then, when you introduce Cre recombinase, you can then cause these two to bind together and loop out and cut out everything that's intervening between those two sites. So then this is the resultant result after you introduce Cre recombinase to the LOX-P floxed mouse. So you see exon 1 is missing. And exon 1 is really, really important because it has a number of features that allow VHL to be expressed and function. So first of all, it has a start site. So this is the part that tells the gene, start making a protein here. The next thing is it has regions that are involved in tumor suppression and in where the protein goes in the cell to do its job. So you can see if you get rid of exon 1, you get rid of a lot of the important functions of VHL. And most importantly, you get rid of the ability to make the protein. So down below here, I have shown you a Pcr based results where we had to do a nested PCR to show, because this is a very large region. So we did 2 sets of PCR to show whether or not we got excision of the VHL gene in our retinas of our mice that got of our flox VHL mice that got Cre recombinase. So they either got Cre recombinase or did not get Cre recombinase. So if it does not get Cre recombinase, this is just too large a region to do PCR, and you get no band. Right? This is just a background band. If you give Cre recombinase, we can now PCR across this flox site and see that we have, when you get Cre recombinase, you get recombination. It's important to note that with these plus ones, we also would have a lot of cells that have the normal allele. You just won't see a band. But we definitely get recombination in our mice that get the Cre recombinase. So now all we have to do is use these flox mice and give Cre recombinase, much like people did in all of these studies back here, right? So what these studies used was a promoter that directed expression in a very specific type of cell, a single type of cell, or maybe 2 types of cells. But that was the approach that they used. We used a very different approach, and I'm calling it an agnostic approach, because we knew that people had tried this in many different ways, and you could try it again targeting vascular endothelial cells or microglia, but we didn't know if we were right about it. We also didn't know if you only needed 1 cell type or if 2 cell types have loss of VHL, right? So from the pathology in humans, we thought that the vascular endothelial cells do not need loss of VHL, but maybe those cells that become the giant foamy cells do. So not knowing and not wanting to put our money on 22 red, not wanting to bet on a single cell type, we wanted to use a more general approach. So we used a viral vector to deliver Cre recombinase. So this means that we can deliver it to a specific tissue type, so the retina. So we can deliver the Cre recombinase specifically to the retina, and this avoids all of the pathology that occurs if you do this to the whole body, or to the brain, or to the kidney. So here's a schematic of an eye. This is cornea, the lens. This is a mouse eye. They have really huge lenses. People have much smaller lenses. There's the optic nerve, and this is the eye socket and the cup back here. So I'm depicting the choroid that contains all of the blood vasculature in red, and then the retina and RPE in blue. Remembering that we also get a vascular supply kind of here, but it's small. It comes through the optic nerve, and these vessels come and feed the retina from the top and the choroid from the bottom. So because the hemangioblastomas tend to be superficial, we injected Cre recombinase into this vitreal space. So this is just a gooey gel-like substance that attaches the lens to the retina, and just it doesn't really attach it, but it's connected to. So when your lens moves, the vitreous moves a little bit. So it's a fluid gel-like substance, which means that you can inject a liquid in there, a liquid that contains a virus, and that virus can then infect retinal cells on the top, and the vascular cells on the top, and the microglia that are in the top. And in fact, this particular virus that we used, AAV2, can penetrate the entire retina. So now we've delivered it, and it can not only can it penetrate the whole retina, but it has general retinal tropism. That means it can infect all of the retinal cell types that have been looked at. Okay, so now we have the ability to cause VHL excision of the flox VHL allele using Cre recombinase in any and all of the retinal cell types. And to then not further restrict expression, we used a general promoter. So rather than using a rod-specific or an RPE-specific promoter, we just used a general promoter, CAG. And that just also, not only can the virus then infect any cell type, the gene delivered in the virus can express in any cell type as well. We also combined this with EGFP so that we could visualize and validate that the virus was being expressed at early stages. So, so we did this and we thought, let's just let the mouse then develop tumors and see what happens. Does it look like a retinal hemangioblastoma? And when we started this study, all of the growths that had been seen by directing expression of Cre recombinase to delete VHL in different retinal cell types, none of them developed tumors that are reminiscent of a hemangioblastoma. One paper recently showed that, and it is very similar to retinal hemangioblastomas, but lacks some characteristics. So, okay, so we did this. We injected this virus. I'm showing you a picture in green showing the reporter for the viral expression, EGFP, and on the right-hand side is just the nuclei of the top-down view of the retina. So these are mostly, this is retinal ganglion cells, vascular cells, intermediate neurons. So we had to use a pretty high amount. These are viral genomes to get expression. You can see if we go twofold, 10-fold, 100-fold lower, that we don't get expression of it. And so we got it to express, so we knew what amount to use. And then so this obviously we took this retina a few days after we expressed it, and then we waited for the mice to develop, and we could follow them in vivo. So one of the things that we know is that VHL hemangioblastomas end up with abnormal vasculature. They have leaky vasculature. They have large leaks, small leaks. They just grow these retinal tumors that are highly vascular. So this is an animal fluorescein angiography, fluorescein retinography. So we can see here the large vessels coming out of the optic nerve. So these are supplied through the optic nerve, and these spread out radially through the whole retina, and then dive down into smaller vessels that feed the intermediate and top layer of the retina. We found a lot of localized leaks shown here and a possible anastomosis. And then we also saw some areas of large leaks. So these would potentially be reminiscent of the exudations and the hemorrhages that you see in patients where they need to come in and get cryotherapy or laser treatment to treat these. Okay, so I jumped. So what are these? I jumped a little too fast there. Okay, so that's a very macroscopic level of what's happening in the retina. What is happening at the cellular level? Exactly where are these abnormal vasculature, these hemangioblastomas located, and what do they look like? So this is a mouse histological section. On the right-hand side here's a normal VHL floxed mouse. And on the left is one of these mice that had a large visible tumor on in vivo imaging techniques. We did others more than just the fluorescein retinography. We did OCT, we did visual looking with ophthalmoscopy, and we were able to see these even by eye, like you go into an eye doctor and they put a big lens in front of your eye and look at the back of your retina. So I'll just tell you what we're staining these with. So DNA is in blue. So these are the nuclei. Again, on the, so this red, well, let's just talk about the B section right now. So the small red stuff at the left-hand side, upper left-hand side near the B, that's the choroid. And that's just where the vasculature is. That supplies the bottom part of the retina. Here are the nuclei. Those are the rod and cone photoreceptors of the mouse. They're very condensed small nuclei, characteristically very bright. And those are the photoreceptors. And then you move to this intermediate layer of neurons, and that's the middle layer. And then up here, it's a little dim just because the rods are so bright, but there are nuclei that are the retinal ganglion cells. So I already told you about those in a previous image, and I can answer questions, or you can view this again if you're not sure what these all are. But these retinal ganglion cells, their axons make the optic nerve that then goes to our brain. Okay, so DNA is in blue, so that tells you where the nuclei and the cells are. We stained this also in red with IB4. It's just a really easy marker for vascular endothelial cells. So here in the normal eye, you can see here's a small blood vessel here with the asterisk. There's another one here. And this layer, this superficial layer, it has the ganglion cells, the vasculature, nerve fiber layers, the axons. So it has a lot of blood vessels across it. But when you look at it in cross-section, you don't see them very often. Right? And then also down here, you can see these vessels are larger, and what they'll do is they'll divide off and they'll dive down into the intermediate layer and feed up until this section between the two layers of neurons. So you can see little red sections here. So that's what a normal mouse vasculature looks like in the retina when you look at a cross-section of the retina. The last stain we used here was microglia. IBA1 stains for microglia, and we looked at this because we know the giant foamy cell pathology in humans, and these cells have the potential to be that giant foamy cell, but they also have the potential to respond to a lot of disease and damaged states. So this is the nucleus of a microglial cell, and you can see its processes. So it has the soma of the cell is located. They're located in the ganglion cell layer as well, but you'll find that the cell, this is probably just a quiescent cell. It has processes that it extends out throughout the retina. Here's another one, maybe from that cell, maybe from another one. And it just keeps track of the health of the retina, what's going on, so that if some damage occurs, it responds, and it can respond quickly. So that's the normal eye of a mouse. When we look over here at the VHL mouse that received AAV2 Cre recombinase, so it's now lost excised VHL, and I showed you that with the nested PCR results, we see a very different picture, and we see something that nobody ever wants to see in the retina. So now we see normal photoreceptor layers, normal intermediate cell layers, but we start seeing on this ganglion cell layer, so on the superficial layer, which is reminiscent of retinal hemangioblastomas in people, we see this very large, very abnormal blood vessel. So it has multiple layers of vascular endothelial cells, which is just not seen. It also has a lot of microglia, so you can see a lot more cells and a lot more reactivity in this area. So the microglia are definitely activated. And then there's this little batch of cells here that stain both for microglia and for vascular endothelial cell markers. So at this level, we don't know if this is an intermediate cell type that has both those markers, or if it's vascular endothelial cells that are being engulfed by microglia in order to try to get rid of them because they're damaged, or if the microglia are here and they're secreting things that promote angiogenesis. We can't tell that from this, but these are all ideas and hypotheses that are brought up by this type of pathology. Okay, so it was, so we also took a light image through this. So this lumen of the vasculature, because there were so many layers of cells, was this a normal blood vessel? Was it carrying blood? And in fact, when you look at it with DIC overlaid here, you can see these little dimpled cells. These are red blood cells. So this is a fully functioning, fully active blood vessel that we're calling a hemangioblastoma because there's this, I'm going to just move back here because it's a little easier to see. There is proliferation in this site. There are nuclei, there are microglia, there are phagocytic cells, and there's vasculature. So this is, besides it being a mess, it's very reminiscent of what we see in the late stage pathology where you have an immune cell, vascular, and proliferation involved in this tumor formation. So we, of course, looked at a number of other features because we wanted to characterize what were the important characteristics that we were seeing. So stuff that I'm not showing to you here, we looked with indirect ophthalmoscope, so we can just look in a mouse's eye, a live, awake mouse and look at the back, dilate its pupil, and then look at its retina. So in about half our mice, we saw lesions. They were either vascular, and they were quite varied, either vascular or avascular. They could be flat or elevated. So there's a lot of different things that can be going on, and they're different. So this could represent potentially different characteristics of the tumor or different stages of the tumor. We also used fluorescence angiography, retinography, and we saw intermittent and chronic lesions. We looked at these same mouse across time. We saw it in about a third of the mice that we looked at. So the fact that they're intermittent means that if we looked more frequently, we might see it in more. We also did OCT imaging, which gives us a picture of the retina, both from a top-down view and a cross-section, and we saw a lot of infiltrations, inclusions, altered thickness, fluid-filled cavities, and disorganization of layers across development. So we looked at these mice at 2, 4, 6, and 8 weeks after we injected them. We found disruptions by OCT in all of the animals that we looked at, and what this basically means is that this is a lot of these things are probably due to the manipulations that we did, doing the injections, maybe putting a virus in, but might not have to do explicitly with the loss of VHL and tumor formation. So then we did histology, where we saw, out of 11 samples, we saw abnormal vascularization, and microglia or a macrophage, so the marker can't dissociate between the two, so and then growths. So why is this number so low? Why isn't it half of them, like with the indirect ophthalmoscope? So as I mentioned, we looked at these mice at 2, 4, 6, and 8 weeks, so we wanted to follow them out. We found that we didn't really see abnormal histology at 2 weeks, but we started to see abnormalities at 4 weeks, and we saw those hemangioblastomas at 6 and 8 weeks. So it definitely takes time to develop. So it takes time for Cre to express. It's expressing by 3 days. It might then take a little while to get the VHL excised. And then once the VHL excised, those cells that are going to become the tumor need to start changing what they're doing and causing the tumor to form. So again, like the human pathology, this histology is once we know that a lesion has developed. So now that we have that, we can maybe go back and look at these markers at earlier stages again. And by immunofluorescence microscopy, I showed you one image of that. 3 out of 6, and these were of the 6 and 8 week samples, showed 4, 6, and 8 week samples. Half of them showed increased number of superficial vessels, and about half of them showed large abnormal superficial blood vessels. So to summarize the data that I've shown you so far, we used an agnostic approach to try to understand the tumorigenesis of VHL hemangioblastomas. So we used a general promoter, a general virus that can infect all sorts of cell types, and we injected it just into the eye into the retina of the mice to avoid systemic complications. We were able to see functionally and structurally abnormal vasculature, and this abnormal vasculature developed with active microglia within 6 weeks. So now we are using this mouse model to further our studies and look at the role of vascular endothelial cells and or microglia, and their and whether or not they have an integral role to hemangioblastoma formation. So I showed you those retinas in cross-section, but that you really don't get an intuitive idea of what that huge hemangioblastoma, what it was doing, where it was going. And we can look at that on the fluorescein angiography, but that kind of isn't very high level of resolution, so it's difficult to get a good view. So we've been working out ways to look at it on a bigger level. And one of the things that we hope to be doing in the near future when COVID allows us to get back to working as normal, is to do some imaging where we can look at the whole retina. And then also, this is a flat mount showing the vasculature at a low magnification and a much higher magnification. So this is just a normal mouse, but when we get back to looking at our mice and looking at the tumor formation, we'll be able to look at this at stages along the way and be able to see whether or not we are seeing abnormal vascular formation early or microglial activation. We'll be able to do this with microglial markers as well. So, we want to do a more intuitive characterization of the abnormal vasculature. And then we're going to directly address the questions of what's happening in the microglia. So as I mentioned before, Wang et al. Put out a paper showing a hemangioblast precursor, if altered for VHL, causes tumorlet and vascular changes at the fundus level that are consistent with VHL hemangioblastomas and some microscopic small lesions, but not the size and level of hemangioblastomas that we see in our mouse model or in patients. So we're going after the microglia angle, and we think that it could be involved in tumorigenesis. Either those cells themselves can become tumorous, like in a glioma, or they might be critical to allowing the tumor to form. So we're going to use microglia expressing Cre recombinase. So now instead of an agnostic approach, we're going to do a very directed approach in our floxed mice. And we can do this with a genetic promoter, just directing microglia to express in a combined mutant mouse. Or we can take a microglia expressing reporter mouse and just look at what happens to those cells when we used floxed mice with the viral Cre recombinase. So the model we already have, but to use microglial expressing EGFP so that we can follow those at earlier stages and possibly follow those in vivo if we have good expression. So we're going to look at what happens at microglia when they lose VHL. We can do this both in vivo and in vitro, so in cultured cells. And then we're going to ask the question is, are microglia necessary and sufficient or sufficient to contribute to pathogenesis? So are they tumorigenic cell type or do they contribute? So we can genetically and pharmacologically get rid of microglia, and we can follow tumor formation, and we can follow fluorescently tagged microglia. And then we can look at what pathogenesis occurs when we get rid of microglia and when we get rid of VHL and microglia. And we'll be able to do this using some more of those intuitive imaging mechanisms that I showed you above. So, so those are our plans of what we are really dying to do, because we just received our funding a few months ago. We've got our mice in our colony, and we've got some new tools available to us, and we're just waiting to get back to work so that we can start making these new variants in our model and test our hypotheses. So I would like to just comment again. I mentioned early on, I worked with Michael Gorin at the Jules Stein Eye Institute. He is a clinician physician, MD, PhD, who has been instrumental in the hypothesis development and experimental development in this early stages. Sachin Parikh, who is in our laboratory. Our Jules Stein core facility allows us to do in vivo imaging. That is run by Steve Nusinowitz, and who is director of the lab, and Eduardo Araujo. And then last but not least, I'd like to thank the VHL Alliance. We had a small grant a couple of years back that allowed us to develop this in vivo mouse model with great success. And I now have continued support for follow-up studies to test the hypotheses. And with that, I'm going to go back to take that screen off and hopefully go back to you seeing me and answer any questions that you have. I think Josh will handle those questions, and to say that we're really excited about this because I think we have a novel hypothesis that has a lot of possibility in either being the tumorigenic cell type or in helping to develop the tumor in the very early stages. Not only that, but there's right now a clinical trial going on that would target hypoxia induction and vascular endothelial cells, but there are other drugs that are in clinical trials that can act on microglia. So if we happen to find that the two together are really what promotes the tumor formation, we actually have some potential therapeutic approaches that we can test in mice, and that potentially might be of interest to developing to human use. So with that, I'll go back to the split screen with Josh so that we can answer questions.
Dr. Matynia, thanks again. That was great. There are a number of questions here, but let's start with a nice lighthearted one from the group. How on earth did you get a mouse with dilated pupils to look you into its eye?
Well, so mice are really small, so we can actually scruff them. So if anybody has a pet mouse or a pet rat, you can pick them up and you can hold them. Mice don't really, they kind of know that they're lunch for many people, so they don't like being held by people that much. But just like a cat, you can, like a little kitten, you can just like grab at the neck behind the head of the mouse. You can do that, or a cat or a kitten or a puppy. You can do the same thing with a mouse, and you can hold it and you can pretty much immobilize it. We use atropine or other dilators that they use in clinic. In fact, we use the exact same thing they use in clinic to dilate the pupil. And we try not to look in there too long because the bright light is annoying, just like it is for us. But it's, so we just have to hold them. You can also do it for all those imaging. We actually have to anesthetize them. So because we need them to not move at all, because that's magnified. But for just looking with the ophthalmoscopy, you can do that with hand holding them.
How did you become interested in research into the retina and specifically VHL-related tumors?
That is too long a story, and I did talk a very long time, so I'm going to not tell a really long story, but I've always been interested in science and biology and disease-related aspects of it. So I have a background from yeast to blood clotting to learning and memory, mice, human, people. So I ended up working in the retina because it is, one, a really cool tissue. It is really important. We highly value our vision and our retina. And I was interested in problems of retinal diseases that currently don't have a good treatment for them. So I have other projects as well in different diseases, but working with Mike Gorin, who sees these VHL patients and has seen them for a long time, he gets very frustrated when he can't treat a patient in a way that really enhances their life and prevent and just manages a disease. So with VHL, all you can do is laser off the hemangioblastoma or cryo it off, and that works, but it can leave scars and it can lead to loss of vision. And so if we can find a way to mitigate those problems or prevent them or slow them, then that actually really enhances someone's life and capability to work. So that's how I got interested in these things. So it's really great working with a clinician because they know what patients are going through and they can tell you things that you would never hear. And if you just read it, it doesn't have the same impact. So that's how I got into this.
That's a great lead into the next question, which is, what are the potential implications of your research on the lives of VHL patients and their families?
Right. So, our hope is that with this mouse model, we'll be able to directly test questions related to what cells are causing the tumor, and with that, get at what can be done to slow the tumor formation or prevent the tumor formation. Now, those are really big goals that will probably take a while to get to. But if we're constantly keeping that in mind, as I mentioned at the very end, there are therapeutic trials going on right now that are for different types of tumors. And if you can use if drugs come out of that, and if they can be used in combination because you find that endothelial cells and microglia are important, then maybe we can look at using drugs that by using combination therapy, sometimes you can lessen the effect, the side effect of a single drug, or you can improve the efficacy. So that's our hope, is that we can get to some of this information in a relatively short time frame, and by that I mean, a couple of years, not a couple of months. These things just don't move that fast. And get some insights that we can then share with others, to look at whether or not we could have potential therapeutics that can be safe in people, and then work with others that can do those types of clinical trials.
Thank you. Here's another question from the audience. Can you speak to the likelihood that multiple treatments lead to other retinal issues?
I believe they have a little bit more down there. I have more than 50 treatments, and as a result, so I can't speak greatly to that. What I can speak to, because I'm not a clinician, so I don't see it, so I know some. So the treatments, as I mentioned right now, are to laser off. So if the hemangioblastoma gets too big, or if it leaks or bleeds, hemorrhages or exudates, you can laser treat it. So you basically cauterize it with a laser, which is pretty easy to get to in the eye. That's one benefit of it being in the eye versus in the CNS. But it can leave a scar. And the exudation and bleeding can cause retinal damage that causes a bigger scar than just the tumor would form. So if you have, most patients have one hemangioblastoma, but many, many patients have 2 or 3 or 4, and some patients have a lot. And so you have these all over the retina. And the more you have, the more likely you're going to have vision loss because of the scarring. So when you laser something or cryo-treat it, you're killing the cells, not just the vasculature, but the cells around that area, because you can't just get the little bit of the vessel that's leaking. So you have to do like a larger spot. So it does kill it. And so you want to treat these when they're as small as possible, or you want them to not leak and you want them to not get big. So there's a lot of damage that can happen depending on the tumor that you have and how often it leaks. So you can laser it, it can leak again. So you have to be constantly surveyed and constantly imaged and go to your eye doctor to make sure that you don't have a new hemangioblastoma, or your old one isn't now exudating. So that's if you kill cells, it's really hard to get them back. So our goal is to try to do something to prevent these cryotherapies and laser therapies to happen in the first place, or to reduce the number of those that have to happen. Uh-oh, no sound.
Here's another question we have from the audience. Are there any links between hemangioblastomas occurring in one location and then occurring in another?
Okay, so that's a great question. And the obvious link is that you have to have loss of heterozygosity at both locations. So we know that most patients that get retinal hemangioblastomas, they get 1 or 2 or 3. To have 10 or more is pretty rare, but it does happen. So you definitely do get them in more than one location. We also know that a retinal hemangioblastoma is very similar to a CNS or cerebral hemangioblastoma, both in terms of the pathology and in terms of the type of tissue, right? The retina is the CNS, so it has all of the same elements that the brain has. And definitely patients get both of those. One of the benefits of the retina is that you can see these early on. So you can, if you get these, or if you have VHL, you should be looking for cerebral hemangioblastomas. There are some interesting theories, and if you listen to Dr. Gardy's talk the other day, she discussed migratory cells and mesenchymal cells that can migrate. So a question would be, is there a single founder that divides and migrates to different locations? Or are there multiple founder cells that become tumorigenic in different tissues? So it's weird to think of a cell migrating from the retina to the kidney or vice versa. That would be very weird. The only thing that could potentially do that would be in your circulatory system, so a cell that can actually go through your blood vessels. So that's So I think that the link between them is more the fact that VHL is a tumor suppressor. So when you have one loss of VHL, you're going to have perhaps a decrease in your ability to ensure that you don't have tumor formation. And then when you have loss of the second allele, that is what allows the tumors to occur. So how does VHL get lost in multiple sites? And that has, because there's very specific target tissues, that probably is a little different in the different tissues. So the microenvironment of the kidney is different from the microenvironment of the retina and the brain. And you don't get tumors of the skin or of skeletal muscle. So there's clearly factors in those areas, and it has been suggested that it's the high need for oxygen. And that's a really great hypothesis. The retina, the kidney use high levels of oxygen. The skeletal muscle does too, but it's not, it's sporadic. So the link is really the VHL gene. And I think what I'm seeing is that we're starting to come across or come together with some ideas about how this could be, how this could generate in multiple different tissues at different times in the development of the disease, or at different times at different ages. So the retinal hemangioblastomas, I think, have been first identified in like 2-year-olds. So it can occur very young. So I think that we're kind of thinking about those things, but they're theoretical at this point in time. But it's loss of VHL is what the link is. How does it happen in different areas?
Thank you. And then we've got two last questions. Why should people outside of the VHL community care about and support your research?
Okay, so again, right, obviously the VHL community has a very vested interest in this, but outside of the VHL community, the research that we're doing and that other people in VHL don't just pertain to VHL, right? So in the kidney, clear cell renal carcinoma is a kidney cancer that many people get. Retinal hemangioblastomas are pretty specific to VHL, but there's still a tumor cell type, and there are other types of hemangioblastomas that occur in people. So what we're doing is we're using VHL to create this model of a tumor, but the questions we're asking are about the basic biology of the tumor. And tumors might involve different cell types, but they have a lot of common needs. So the vascularization, as I've discussed already, what we know about the ability to deny vascular feed to tumors came from a lot of research many years ago, but it will also apply to these tumors of VHL. So what we find in the VHL tumors is highly likely to also apply to other types of tumors, whether they're benign or cancerous. A lot of the biological processes that are occurring are going to be relatable to other tumors. The fact that we're looking at microglia, CAR T cells in cancer right now are a really big story. How do you modify immune cells in order to beat certain types of cancers? So we have an immune cell that's innate in the retinal tissue. So maybe these innate cells, immune cells, also have a role in other types of brain tumors or CNS tumors. So, we are looking at both what's known in other types of tumors and where you can find out in our types of tumors that might inform other tumors. So it's also fairly predominant disease. So it's definitely something where we want to try to, basically we have health issues, and this is an unanswered disease that we can't really treat very well. We are treating other causes of blindness, so we want to have, improve our ocular health, and some of the treatments that we find for VHL-based tumors might also apply to other types of tumors in the eye, like retinoblastoma. We don't know that at this point in time, but these things are not solitary diseases. They have commonalities.
Thank you. And we have one last question. The older you get, do you have a less chance of developing retinal hemangioblastomas in VHL patients?
Well, what I understand about the tumors that occur in VHL is that you have various ages of onsets, and they've done natural history studies. My VHL is a natural history study of the tumors and the symptoms and phenotypes that patients have. Phenotypes are more for mice and people, so the symptoms that people have. And unfortunately, what happens in VHL, because it's a tumor suppressor, and the older you get, the more chances you have of getting tumors. So if you have loss of that tumor suppressor, the older you get, the more chance you have. So as I said, the youngest is 2. Most people get diagnosed with VHL in their 20s or 30s because they have tumors that form. But you can have a tumor form anytime in your life. So you have to be continuously vigilant about screening for it. The older you get, it's not less likely that you get a tumor. The older you get, the more likely it is that you will have one or a second one.
Dr. Matynia, I just want to thank you again for taking the time to talk to us today, as well as all the important work you're doing in VHL research. It means so much to us. Thank you very much.
Well, thank you for letting me talk on for so long.
We appreciate it. Thank you.
Take care.
And for all those watching at home, you can find more Meet the Researchers as well as other events in the Surviving and Thriving Together series at vhl.org/together. That's vhl.org/together. And until next time, stay safe. Dr. Matynia, thanks again.